Dear students, welcome to Tunga online classes. Today I am going to start with the proteins. So there are different structures are there in protein, right? So three-dimensional structure of proteins that we come across over here. So those structures are primary, secondary, tertiary and quaternary structures of proteins. They are set as. Okay, there are mainly four structures of protein that we come across, right? So one by one we'll discuss today now, right? In the last video we were discussing amino acids. Amino acids are the building blocks of the proteins, right? Amino acids are bound together with the help of a bond called as peptide bond, and with the help of these peptide bond, so amino acids bounded to form polypeptide chains. Those polypeptide chains are nothing but they are proteins. Let us start with the structure of proteins now. Structure of proteins. What are the different structures of proteins that we come across? Our first one is primary structure of protein, and second one is secondary structure of protein, third one is tertiary structure of proteins, fourth one is quaternary structure of proteins. So these are the four major types of structures of proteins that we come across over here. First one will start with, what is this primary structure of protein? You may ask here. So first one will start with primary structure of protein. Okay, and how this primary structure of protein is synthesized, let us see here. Primary structure of protein is a type of protein in which it has simply the chain of amino acids which are bounded with the help of the peptide bonds itself, right? So this is the simple structure of the proteins, that is primary structure of protein, and this chain of protein is formed as a result of transcription as well as a translation, we call it as. What is the transcription? Already you know. So biosynthesis of mRNA on one of the strands of DNA is called as transcription, and simultaneously the mRNA is decoded into amino acids, that is amino acid chain, so that forms the protein molecule, right? So that is nothing but translation, we call it as. The detailed study of this transcription as well as translation you are going to learn in molecular basis of inheritance, is one of the chapters in PUC2, right? So that is why, so the detailed knowledge about this translation and transcription is not needed to explain over here. So you simply know, so these are the two processes through which the protein synthesis is taking place, right? So let us see. So this is the simple structure of a protein which contains the chain of amino acids, so that forms the protein molecule. That forms the protein molecule. It is formed as a result of transcription and translation, right? So these are the two steps of protein synthesis. So these are two steps involved in protein synthesis. Okay, so this is nothing but what? So primary structure of protein, right? Sanger is the name of the scientist, the one who determined the structure of the primary primary structure of insulin. He determined the primary structure of insulin. So this insulin is made up of, it is made up of 3 chains, right? How many chains? So 3 chains it is made up of. One is alpha chain, and second one is beta chain, and third one is connecting chain, right? So alpha chain, so which contains 20 number of amino acid. There is a chain of amino acid, right? That forms the protein molecule, right? Alpha chain is made up of protein molecule that contains 20 amino acids, right? Simultaneously, so these 21 amino acids, and this one beta chain that contains 30 amino acids over here. So total number of amino acids present over here are 84. Then how many remained back? They form the connecting chain. Actually, the active molecule of this insulin is made up of only 2 chains. One is alpha chain as well as beta chain. These 2 chains are joined with the help of connecting chain over here. So the total number of amino acids is 84, right? But actually the structure, active structure, so that contains only 51 number of amino acids. Why? Because it is made up of only alpha as well as beta chains. Okay. So this is about the insulin structure of insulin. See here, this is how the structure of insulin is. So this forms here to here itself forms the alpha chain, and this is the connecting chain, C chain we call it as, and this is the beta chain. So beta chain like this. So there are 3 chains that we come across in the molecule of water. So proinsulin gene it is called as proinsulin itself, right? And this connecting chain is removed and these 2 are joined over here so as to form insulin molecule. Alright, so this is how the insulin itself is consisting of a chain of amino acids. A chain of amino acids. This is the primary structure of protein we call it as. And the next one is secondary structure. So what is this secondary structure? So how it is and what are the forms that we come across here? Let us see now. Next one is secondary structure of protein molecule. Secondary structure of protein. So this is secondary structure of protein, which becomes a 3-dimensional helical structure, right, which is made up of a chain of polypeptide itself, right? So it is also made up of polypeptide chain. A polypeptide chain makes a helical structure. Over here. So this helical structure, which is of different types. Here alpha, and second one is beta, and third one is collagen helix itself. It is called as collagen helix. See here, alpha, it is also helical structure, and beta is pleated in the sense that it makes the sheets of the polypeptide chains over here. So alpha helical structure or alpha helix, beta pleated sheet, pleated or sheets, and collagen helix. So these are the 3 kinds of structures that forms the 3-dimensional structure of the secondary structure of protein over here, right? So let us see. So these helical structures are maintained by this structure, I mean secondary structure of proteins is maintained by intra as well as intermolecular. So see here, intra and intermolecular connections has been done by the hydrogen bonds. Hydrogen bonds means here intra means within the molecules and inter means in between two molecules, two polypeptide chains. So there the hydrogen bonds are linking these one so as to form this secondary structure of what? So structure of protein itself. Say for example, so this is one polypeptide chain and this is another polypeptide chain. These two are held together with the help of hydrogen bonds over here. So this is intermolecular, in between two molecules of polypeptide, right? So two polypeptide chains it is said to be as. In the same way, within the same polypeptide chain, the bonds are found over here, right? So those bonds are what? Hydrogen bonds. So this secondary structure of protein is maintained by the intra as well as intermolecular connections called as hydrogen bonds over here. So with the help of hydrogen bonds, they have been bonded to each other. So let us see in alpha helix. In alpha helix, so a single polypeptide chain makes a helical structure. A helical structure over here. So a helix of a single polypeptide chain has been made over here. So best example for this one is the keratin, which is present in the hair itself. Hair keratin protein itself is the best example for alpha helix over here, right? And myosin in muscle fiber, it is present over here. Keratin is one best example, myosin is another best example. These two form the alpha helix secondary structure of protein over here. And beta pleated or sheet that we come across over here. Pleated structure or sheet like structure that we come across over here. And how this beta pleated sheet is made? So it is made up of what? Made up of two or more polypeptide chains. It is made up of. So it is made up of two or more polypeptide chains bound together with the help of what? Bound together with the help of hydrogen bonds over here. Hydrogen bonds over here, forming a sheet-like structure over here. So this is beta pleated sheet, what we call it as. And last one already I have mentioned, so alpha helical, beta pleated as well as collagen helix. Third one is collagen helix. See here this collagen helix, collagen fibers, white fibers that you come across over here. And so this collagen helix itself is made up of 3 number of polypeptide chains coiled each other, and both So all the three are interconnected with the help of hydrogen bonds over here. Imagine the three-dimensional structure of this, so collagen helix. So here three helical structures are there, and all the three helical structures are coiled each other, right? So all of themselves are interconnected with the help of hydrogen bonds, and that fiber it forms what? Collagen helix itself. It is made up of three polypeptide chains. Three polypeptide chains here. It is made up of three polypeptide chains which are coiled each other, to form three-dimensional structure of protein, right? All the three are helical polypeptides. All the three helical polypeptides are coiled each other so as to form one helical structure, that is called as collagen helix, and all the polypeptides are linked in between themselves with the help of hydrogen bonds, so that forms the three-dimensional structure of secondary, so structure of proteins over here, right? So this is how there are three types of structures that we come across in secondary structure. One is alpha helix, and second one is beta pleated one, and third one is that is collagen helix, right? So these are the structure of secondary structure of protein molecule. And let's go to the next structure, that is tertiary structure of protein now. Yes, now tertiary structure of proteins. Tertiary structure of proteins. See here, this tertiary structure of proteins itself is nothing but the secondary structure of protein folds or coils and folds so as to form very compact structure over here, and that compact structure with the functional units called as a tertiary structure of the protein molecule. Let us write. So this is a type of structure of protein which is made up of coiled and folded secondary structure of protein to form a compact structure of protein with functional units. So see here, this protein molecule that coils and recoils so as to form one compact structure over here. So it forms a compact structure over here, and this compact structure which is formed itself is said to be as what? Tertiary structure of proteins. It is a tertiary structure of protein itself. And so the biological activities what are going on, right? All of themselves, biological functions what are going on with the help of proteins, it is being done by the tertiary structure of protein only. So this is biologically very important, and it is functional protein. It is a functional protein over here, right? So this is more important point to be noted. And here some various number of bonds that are involved in formation of this 3D structure of three-dimensional structure of the protein molecule over here. So this is said as 3D structure of protein molecule over here. So that is three-dimensional structure of protein, and this three-dimensional structure of protein itself is called as native state. It is said to be as native state. So native state is nothing but three-dimensional structure of protein itself. And this tertiary structure was first made by identified and explained by Kendrieu as well as Perroge are the two scientists in the year 1963. So point to be noted. So they discovered what this tertiary structure of protein, and its 3D structure of protein itself is called as a native state, and this is made up of this is made by 5 types of bonds over here. So 5 types of bonds are involved in making this tertiary structure, and those bonds are ionic bond. Second one, hydrogen bond. Third one, van der Waals. So interaction it is said as, right? Hydrophobic interactions, phobic interactions. So like this, ionic bond is there, hydrogen bond is there, van der Waals bond is there. So hydrophobic interactions are there. So different types of bonds are involved in binding or in formation of this tertiary structure of protein. See here, these bonds, one more I have to write here. So disulfide bond. So these bonds are easily broken with the help of some of the factors like high temperature. When high temperature is given, when pH is more over here, high temperature is given, that moment what happens? So the 3D, three-dimensional conformation of the protein molecule that breaks up, right? And this process of breaking of the tertiary structure or the 3D structure of the protein molecule is called as denaturation. It is called as denaturation of protein. It is said as. So it is also acidic nature. So many factors involved in that, right? So like this. So denaturation of protein molecule that take place because of all these factors itself, right? So that's why, so the biologically very important structure of protein is nothing but tertiary structure of protein only. So this is the fact what you must remember over here, you must stress over here, right? So I hope you understood. So what is the primary structure, what is the secondary structure, what is the tertiary structure of proteins now itself. And let us start with the quaternary structure of proteins. Let us start with the next structure of protein, that is quaternary structure of protein now. Fourth one, quaternary structure of protein molecule. Quaternary structure of protein molecule. So how is this quaternary structure of protein molecule? It is also made up of 2 or more number of polypeptide chains. So it is also made up of 2 or more polypeptides. polypeptide chains itself. So that's why they may be oligomeric or they may be multimeric in structure. It is said to be as, right? So oligomeric or multimeric means many number of polypeptide chains may be there. So minimum number of polypeptide chains may be present over here. So all the polypeptides have polypeptides have primary, secondary, tertiary structures are present over here in all these one. And if polypeptide chains are similar, then homogeneous. So polypeptides they are said as, or homogeneous quaternary structure of protein it is said as, or sometimes they are different one, then they are said to be as, so heterogeneous quaternary structure of protein it is said as homogeneous and heterogeneous polypeptides that we come across made up of similar polypeptides. So these are made up of different type of polypeptide chains itself, right? So this way there are two, so polypeptides they are made up of, it shows there are different types of polypeptides that we come across over here. So these are also bounded with the help of some of the bonds, same ionic bond, hydrogen bond. So like this, all these bonds are involved in binding all these polypeptide chains to form the quaternary structure of protein now, right? So this is also very important aspect to be remembered over here. So and the next one, this quaternary structure of protein molecule, what is formed? So by bounding many number of polypeptide chains, so they may be similar, then they are said to be as homogeneous one. If they are different, then they are said to be as a heterogeneous one. So these are functionally very important one. So how these are functionally very important one? So there are different types of quaternary structure of proteins that we come across over here. Example for this one, insulin is one example which is made up of two polypeptide chain. So and this is bounded with the help of the bonds between themselves, right? So this is more important point to be noted. So example for this one is insulin is one which is made up of two polypeptide chains, right? This is one example. And hemoglobin which is made up of four polypeptide chains, it is made up of these are the quaternary structure of protein molecules, what we call them as. And apart from this one, so structurally these quaternary structure of proteins, so how they are, so what are they responsible for? So in shape these proteins may be, first one, it is fibrous in nature. Say for example, so collagen fibers, right? Collagen fibers are best example for this one. Yellow fibers. So these are fibrous in nature, one thing. And they may be globular in nature sometimes. Example for this one is, so that is myoglobin, myosin, that is myosin, actin. So here egg albumin, albumin is there, right? So not this one. Albumin is one such, and hemoglobin, egg albumin, hemoglobin, right? So these are the best example for globular proteins over here, right? So this is one thing. And functionally these are very important. So these are functionally very important. What kind of functions they perform? So they even help for transport. They help for transport. Hemoglobin is best example for this one. It helps in carrying oxygen as well as carbon dioxide, that is the respiratory gases are being so transported with the help of these proteins over here. So it helps for transportation, and so it forms some of the enzymes. So amylase enzyme is best example. Amylase enzyme is best example over here, and even it helps in so what passage protein as passage protein. Example for this one is best example is glucose. Glucose transporter number 4. Glucose transporter number 4. So is transport protein or passage protein, it is said as, since it is present in the membrane of plasma membrane, right? So and so even it is helping as what? So some others are protective in nature. So these are protective in nature. So example for this one is immunoglobulin. So this is nothing but antibody. They are nothing but antibody molecules. These are also one kind of proteins. Y-shaped antibody molecules are also what? So they are protective in nature. They are a kind of proteins over here. And even these proteins are acting as the receptors even. So these are acting as receptors. So receptor of smell, receptor of sound, etc. So sensing, sensory receptors, these are acting as, so right. So like this in function. So scleroproteins are there. So some others are scleroproteins. Like keratin is there, scleroprotein, it is called as keratin is best example for this one. This way functionally there are different types of functions have been managed by these proteins itself, right? So quaternary structure of protein this is. So by this we finished all the 4 types of structures of proteins, and let us go to so the classification of protein on a different basis over here. There are different types of proteins that we come across over here on the basis of their shape, on their chemical composition as well as their function itself, right? So on these 3 bases they have been classified over here. So first of all we are going to classify these proteins on the basis of their shape, right? So let us start with the shape. Classification of proteins. So proteins are classified on the basis of shape, chemical composition, as well as so one more, that is functions. They have been classified. First of all, we classify the proteins based on shape of these proteins. There are two shapes that we come across over here. One is fibrous in shape and globular in shape. First one, fibrous shaped proteins these are. So these are non-enzymatic one. These are non-enzymatic secondary proteins these are. Secondary structure of protein forms the fibrous proteins, right? Secondary structure of protein makes the fibrous protein. They are non-enzymatic one and they are structural proteins. Means these are responsible for formation of some structures. Example for this one is collagen, fibers, elastin, fiber, as well as keratin of hair. So these are the best example for this one, right? So based on the shape, there are two types. One is fibrous and second one is globular in nature, right? So the second one is globular proteins. So as you know, these are spherical shaped proteins. These are spherical shaped proteins. These spherical shaped proteins may be enzymatic sometimes, enzymatic, and sometimes non-enzymatic, right? And these globular proteins, so they are made up of different structures of proteins like tertiary structure of protein, and even they are made up of quaternary structure of protein they are made up of, right? So this is also more important one. And such kind of globular proteins are hemoglobin. Hemoglobin is best example over here, right? So these are non-contractile one, right? So here albumin is also best example. Here albumin is example. Hemoglobin is example, right? So like this, there are different types of globular proteins that forms different kinds of structures over here. So this is how we are classifying these proteins on the basis of their shape itself. This is one thing, and on the basis of their chemical composition. Based on chemical composition. So there are different types of proteins that we come across over here. So what those are? They are simple proteins. Simple proteins are one thing, and second one is conjugated. Conjugated proteins are second type of proteins, and third one is derived proteins. So like this, there are three different types of proteins that we come across. First one is simple proteins, conjugated proteins, derived proteins. This way they have been classified over here. So first of all, we will start with the simple proteins, right? So first of all, we will start with the simple proteins now. Simple proteins are those proteins which are made up of only the amino acids. And such kind of proteins are said to be as what? Simple proteins. These are the type of proteins which are made up of only amino acids. Such kind of proteins are called as simple proteins. Example for this one is albumins are the best example for this one. Albumins, serum albumin is there, egg albumin is there. So they are egg albumin, serum albumin, right? So they are the best example for this one. Albumins. After that one, globulins. Already I have mentioned these globulins, globular proteins. These are spherical shaped proteins. So these are also best example for simple proteins which are simply made up of amino acids over here, right? And glutenins, they are the stored proteins. They are the stored proteins in, so wheat, right, maize, etc. Right? Glutenin are seen in maize, wheat, etc. Glutenin is there. Prolamines are there. Protamines are there. So these are also simple proteins over here. Along with this one, sclerotic proteins I have already mentioned over here. So sclerotic proteins are keratin is there, actin is there, myosin is there, that is collagen as well as what, yellow fiber, elastin is there, collagen, that is elastin as well as keratin are the best example for sclerotic proteins. These are, right? And next one is histone proteins. Histone proteins are the proteins associated with the genetical material, that is DNA as well as RNA itself, right? So histone proteins are also there, right? So these are all different types of simple proteins which are chemically made up of only the what? The amino acids. And second type of proteins, so conjugated proteins. What these conjugated proteins are, let us see now. Yes, conjugated proteins. Conjugated proteins. As the name suggests, conjugate means join. Means these are the proteins which are joined with somebody else. That is nothing but something else over here chemically. So that something else is nothing but it is non-protein moiety, it is said to be as, right? So and that non-protein moiety is called as prosthetic group. Means proteins associated with prosthetic group it is, or conjugated group it is called as, right? Conjugation group it is said to be as, right? So these are the proteins. These are the proteins which contain amino acids and other non-protein moiety called as prosthetic group. What it is called as? Prosthetic group or conjugation group it is called as. Conjugation group or prosthetic group it is said as. And such kind of proteins are called as conjugated proteins, right? This prosthetic group may be different type, right? Prosthetic group may be different type. So example, different types of conjugated proteins that we come across over here. One is glycoproteins. What these glycoproteins are? So here glycans you know, and proteins. See here, if the proteins associated with the glycans or carbohydrates, so they are said to be as glycoproteins over here. If they are associated with the So what? That is glycans or carbohydrates. They are said to be as what? Glycoproteins. Glycoproteins. The best example for these glycoproteins is, so that is egg white is best example for this one. Along with this one, mucin of saliva. Mucin of saliva. Right, one best example. Egg white is best example. These are glycoproteins, one thing. And nucleoproteins is the second one. So what these nucleoproteins are? Nucleo stand for nucleic acids plus proteins. So together form what? Nucleoproteins. Means these are the proteins associated with DNA or RNA, said to be as what? Nucleoproteins. Especially it is associated with DNA. So as you know, histone proteins. Histones are the proteins which are associated with what? These nucleic acids. They are said to be as nucleoproteins. And the next one is lipoproteins. Third one, lipoproteins. So here, lipo stands for lipids plus proteins. Proteins is proteins. Lipid is prosthetic group. It is, right? So lipids associated with the proteins called as lipoproteins. The best example for these lipoproteins is cholesterol. Its best example, fatty acids. So cell membrane is there, egg yolk is there. So all these are said to be as what? Lipoproteins. High density lipids, low density lipids. So these are also example we can give over here. So these are lipoproteins. And the next one is phosphoproteins. So these are the proteins which are associated with phosphoric acid plus proteins. So together called as what? Phosphoproteins. The best example for this one is casein of milk. Casein is one type of protein present inside the milk, right? So that is phosphoprotein. It is said to be as. Okay, so this is also one more type. And the next one is metalloprotein, right? So next one is metalloprotein. Fifth one, metalloproteins. What is metalloproteins? You may ask over here. So see here, proteins plus metal ions association. Metal ions are the prosthetic group over here. So example for this one, which kind of metal ions? Cobalt is there, Fe is there, Cu is there, right? Magnesium is there. If they are proteins are associated with all these one, then they are said to as metalloproteins. They are said to as. And the next one, sixth one is very important one, that is chromoproteins. What is that? Chromoproteins. What is chromoproteins are? The name itself suggests chromo stands for color. Color stands for pigments. Proteins associated with pigments. Right? So they are said as chromoproteins. Pigments are the prosthetic group over here. right? The best example for this one is hemoglobin, cytochromes, right? So all these are the best example for this one. Hemoglobin, Hg simply B, right? So cytochromes are the best example for chromoproteins. This way, so along with the prosthetic group, proteins are associated, and such kind of proteins are said to be as what? Conjugated proteins. And the last one is the best one, that is derived proteins. What these derived proteins are, right? So they are simply, so the broken simple proteins, right? The denatured simple proteins. If the simple proteins as well as the conjugated proteins are denatured, then what kind of so pieces that remains back, they are said to as derived proteins, they are called as. Let us write it now. Derived proteins. Third one, derived proteins. These are denatured or degraded simple and conjugated proteins, right? So they are said as derived proteins. Example for this one: proteoses, peptones, polypeptides. These are the best example for derived proteins. So these are the different types of the proteins based on their chemical composition, right? So these are the classification of, this is all about the classification of these proteins now, right? So by this I am going to conclude this class over here. So next, that is biological importance or the significance of these proteins is also very important one. We will discuss that one in detailed way in the next class. Thank you.